Добрый день, уважаемые коллеги.
Good day, dear colleagues. Let us, on behalf of the organizational committee, greet all of you and to thank you for participation in the work of our session, Innovative Biotechnologies for Agriculture. We would like to express our gratitude to our guests from Russia, from Belarus, from Pakistan, those who accepted our invitation and who are here to present their works in our session. Let us start our conference. In our program, we have 9 reports. The first report will be presented to us as a video recording. It will not be online, it will be a video recording. And you know, their presentation lasts for 25 minutes, so we will have to show our patience because there is no opportunity to reduce it. However, we have to follow our schedule and the time allocated to each of us. So each of the reporter will have 15 minutes. So we suggest to have 10 minutes for the report and about 5 minutes for questions and answers. But as our first report lasts for 25 minutes, and then before coffee break we have coffee break, so maybe let us have 8 minutes, let's say, for presentation and 3 minutes for questions and answers, if you agree. Maybe then questions could be asked somewhere at the background, after the reports. Yes, during coffee break you will have a chance to ask any questions to our speakers, and that's what we suggest you. Do you accept these time requirements, let's say? If you have questions to our first reporter, you can send your questions through email, and then you will receive the recording. We will have video recording of our first speaker. Okay, the topic.
Лептину Тарса, Аллах Бахш, заведующий лабораторией биотехнологий семян Центра передового опыта в области...
The head of Seeds Biotechnology Laboratory, Punjab University, Pakistan.
Accept my apologies for not being there physically, and at the same time, I'm really happy to convey my best greetings, regards, and congratulations to the team of NCB on reaching remarkable milestone of 30 years of groundbreaking research and innovations. Your institute has been a beacon of knowledge and a hub for fostering scientific excellence. May the next many years bring even greater discoveries, collaboration, and advancements that will continue to shape the future of the country. So today I will talk about how the small RNA molecules can be used as a pesticides, potentially can be used as a pesticides for the crop protections. And I will give you examples from our, case of Colorado potato beetles, which is one of the devastating pests of potatoes. So, as you know, we are surrounded by a lot of challenges. And one of the main challenges we see today is climate change. And the climate change is also affecting the pattern of so many things. And among them are the pests. So according to one estimates, if the average global yield losses from the insect pest will rise to 20 to 25, that's really a big challenge for all the breeders, for all the entomologists, for all the scientists related to agriculture, to breed climate-smart, resilient varieties, to bring in the innovative technologies that can really do wonderful job to mitigate the climate changes and also to tackle the pests. The examples, the case study which I'm showing is related to Colorado potato beetles, how we use the dsRNA to control these CPB. I would call CPBA would be Colorado potato beetle. So if you look at the overall global losses from different pests, you can see the insect pest, weed pathogens, and weeds totals 42 point. It's a great number of yield losses by the farmers, and that's a cause actually that triggers the research for bringing in the new technologies for our agricultural landscape. So for this, so you see, what is the general practice worldwide in order to control all these, all these pests and diseases? The chemical insecticides and pesticides are commonly used. So, but the situation is not really happy. If you look at this slide, also it's a bit old data. The $17 billion, is the amount as the total is the insecticides sale worldwide. That's a huge market actually. But although this is the widely practiced method all over the world, but the problem you see is the development of resistance against these insecticides. And you can see that insect pests, they develop resistance, and approximately 10,000 insects have developed resistance against different chemical insecticides. And likewise, more than 100 weeds, they also develop resistance. And the same is the case with the fungi. And I will come back to GMOs as well. And if you look at the cumulative cases, 586 species of different insects have developed resistance against these chemicals, and have developed resistance against 320 insecticides. Different insecticides have also have been ineffective, less efficacious. So what actually this is the one way to control these pests, like I said, which is commonly used to control. But on the other hand, since 90s, we have been witnessing the changes in the field of transgenes technology, and until now, today, more than 200 million hectares of biotech crops are being grown in different countries. This is considered another effective way to combat these pests. For example, but again, the situation is not very interesting. If you look at this interesting study by the group of Bruce Tabashina, they conducted the study in 5 continents in 2005, and they said that on one side, and you can see that on one side, the hectarage of BT crops is being increased, but on the other side, the number of insect pests developing resistance against these, against these crops is gone to 5. And remember, the study was conducted in 2013. The same group, they conducted another interesting study in 2016. Just consecutively, they kept on monitoring, and you can see that the acreage of BT crops has gone to more than 100 million hectares BT, either as a single trait or in a combination with the herbicide-resistant trait. And you can see on the other side, the number of insect pests developing resistance against these crops has gone to 16. So that's alarming situation. And again, another articles from the same group have been published in Journal of Economic Entomology very recently, and you can see that the number of insect pests developing resistance has gone to 25. So you see, on one side, whether we use chemical insecticides or GMO technologies, we are witnessing the changes in insect pests. They're developing resistance against these BT crops and also against the chemical insecticides. And this basically, the scientists, they say that, well, in order to delay this resistance, you need to adopt different strategies. For example, you have to go for the multiple genes rather than single genes, and try to use the chimeric genes to increase the number of receptors in insect pests of these endotoxins. And also, some people talk about the different promoters, because in GM crops, most of the commercialized transgene crops, they contain 35 as a constitutive promoter. But some scientists are of the view that why not to use the tissue-specific and inducible promoter to confine the expression of these foreign genes at a particular place or at the time of infection. Then will people talk about the different gene expression, and also the field text, different biosafety guidelines have been provided by different regulatory agencies to delay this insect resistance. Refugia is one of them. So, all these strategies are there and being practices, but what is the most important? We need to integrate the existing technologies with also another technologies, the more robust, more eco-friendly, and one of them is RNAi. RNAi basically, is a natural mechanism, and we got the liberty today, and thanks to these scientists, great scientists, Andrew Fire and Craig Millar, who did great jobs and for the discovery of this RNA and got the Nobel Prize. And why today the science has given us this much liberty that we can exogenously express dsRNA that is being chopped by the dicers into small siRNAs, and those siRNAs bind with their targeted miRNA, and hence you can inhibit, suppress the transcript levels or the stop the expression of a particular protein. So, and interestingly, we can use these dsRNAs by different methods. We can, and while in this interesting, I have taken this slide from a review article, and you can see that the red part, it contains the use of these dsRNA that would be considered as a transgenic crop or a recombinant virus such embryon. But on the other side, this blue part, these are the non-GMO, eco-friendly technologies, and you can develop these dsRNA, the tiny molecules, and you can inject into insect body cavity directly. You can use them as a traps, and also you load them with nanoparticles. And the foliar application, this is the area where we are very interested and to develop in developing a bioinsecticides against the Colorado potato beetles. And we can, by the way, you can use by trunk injections, root soaking, soil drenches, seed dressing. So these are all different technologies, the delivery methods of these dsRNA. So we, like I said, that we work with Colorado potato beetle, and Colorado potato beetle is a very the devastating pest of potato, and potato is really very, very important crop of country like Pakistan and Turkey. And Pakistan became recently one of the ninth largest potato producer, and in Turkey is almost producing the same quantity. And one of the main challenge in Turkey of this crop really is the Colorado potato beetle. So why it is so dangerous? Because the adults as well as the larvae of this pest are really devastating. They consume the foliage, and sometimes, like here you can see, that there is no economic yield. All the leaves, foliage is eaten up by the insects, and there is no economic yield. And the problem here is that this insect has developed resistance against all the chemicals available in the market. And why this insect is so small? Because of its active detoxification mechanism. So whenever you can say that whenever the toxin enters in the body, the insect quickly excretes it through its detoxification mechanism outside the body. And also the insects also develops very quickly to different climates. Also, the lack of the predators is another challenge to control this insect pest, and also new host associations. So what we try to do, we try to find out some very, very vital targets from this insect pest and try to develop this dsRNA against the insect. The one we usually develop, we work with the ECR. ECR is the diacin receptor, when one of the very important, the player in insect metamorphosis. And what we tried, we were trying to inhibit the ECR so that the insects may not shift from one larva install to another larva installs. So overall, the molting process was the target to inhibit. On the other side, the other target, for example, in this particular case, we use VATPase. VATPase is also very important, and it maintains this acidic environment, and this VATPase actually facilitates this acidic environments that enhances the activity of digestive enzymes, aiding in the breakdown of digestive food. So these were our two main targets, and the development of dsRNA is really easy, and we got recent very fine sophisticated techniques nowadays. For example, the one we are using is L447. You can just select your gene of interest, like in our case, ECR and VATPase. In this particular case, you can clone it in L440 vector. And the interesting thing about this vector is that the T7 promoter, you can see T7 promoter is there, also the T7 promoter is here. But interestingly, they are in reverse orientation, so that the development of dsRNA could be made, like you can see here in this case, and that can be further induced by IPTG, and this process is very similar. I will not go into the details because we also work with a lot of targets of sclerotic beetles. So I'll focus only with ECR, diacin receptors, and VATPase. So this is the feeding assay. We just tried to see how it really works, and what we were trying to monitor the mortality percentage, larval weight gain, foliage consumption, mRNA. We were trying to see the effect of this dsRNA, the lethal and sublethal effect on mRNA levels of these genes. And what again, we were really interested to see the adults' egg-laying behavior and egg viability. And so these were our treatments. We were using dsRNA with no dsRNA basically, and this was again another control with the dsRNA of GFP, which is the non-target, and we ATPase, USP, ECR, and interestingly, we combine two dsRNA to see whether there is any synergistic effect or not. And this is the consideration of dsRNA we used, and experimental design of CRD, 6 replications were used, and number of the insects were 10 per each Petri plate, and overall, this is how we calculated the mortality. And some of the results, and the results were really interesting. For example, in first instar larvae, you see that approximately 90% mortality of larvae was achieved in case where the larvae were fed with the dsRNA of USP, ultraspiral protein, and ECR. So these you can see that, and you know, in molting, among the targets in molting, we had also one ACR and also USP. These are the molting related targets, and then we just combine these targets, and you can see approximately 90% of the mortality was found, followed by the other dsRNAs. And in control, very less mortality was found. So in second instar larvae, you can see again 90% mortality was observed of those larvae feeding on the leaves, the foliage treated with double dsRNAs, USP and ECR. And likewise, the others, VATPs, and followed by the other dsRNAs. So the interesting thing is that the combination of dsRNA of USP, ultraspiral protein, and ECR was really effective in controlling it up to 70% mortality we recorded with this one. And then in fourth instar larvae, you see why we were using different instar larvae, because we believe that with the increase in larval instars, the insects detoxification system becomes more stronger. And again, in fourth instar larvae, with this one, more than 50% mortality was found with USP and ECR. Still, we believe that this is a good enough one, and we kept on monitoring the lethal and sublethal effects, and see, because we were monitoring the insects. And when the insects were allowed to feed on the treated dsRNAs treated foliage, you can see that the final, for example, in this case, in control, the larvae consumed foliage and its weight gain was, let's say, 75 mg. Well, in case of the one with the treated one, they could not eat too much, they could not consume too much foliage, and the same was the case with other dsRNAs. And this is the case with the fourth instar larvae. Again, you can see even the fourth instar larvae could not consume too much foliage compared to the control. And here you can see significant differences among that. And then like I said, we kept on also monitoring the foliage consumption. We just see that how much the foliage is consumed. Again, you can see compared to the control, the foliage consumption in treated dsRNAs was very, very less. And also in fourth instar larvae, again, the situation was the same. So the larvae were not able to consume more foliage because of the lethal effects, sublethal effects of these dsRNAs. So further, we just checked that whether these we treated those leaves or foliage with the dsRNAs and allowed the insects to feed, and then we tried to see the effect of these dsRNAs on insect body, and that's what we did through we checked by the expression of these genes by qPCR, and you can see if you see the graph, it would be interesting to see that in first, second, and third and fourth instar larvae, the transcripts levels of vacuolar ATPase genes were really reduced compared to the control. Or statistically significant reduction was observed. Well, in case of USP, as a separate, again there was a reduction in the transcripts levels by qPCR assays, and it's similar in case in ECRs. You can see again the reduced transcripts levels of ECRs were noticed. And in case of the one we just combined USP and ECR, here you can see this was really interesting that the transcript levels were really reduced in targeted insect pests. So this also established us that the core RNA machinery really is working within the insect body after the foliage consumption. And this is just comparative images. You can see the larvae, for example, this is the control larvae, and this is the larvae fed with the dsRNA of USP and ECR, and you can see the deformities, the shape of larva. And also the size, if you look at the size, there is a very significant reduction. Similarly, in case of ECR, again, the deformities were noticed in USPs and B80Ks. So this somehow it established that these dsRNA are really having lethal as well as sublethal effects. So this you can see the pupa, and the pupa, the situation is again, if you compare with the control, you can see the very interesting differences in B80Ks and ECR in USP, and also the combination of USP and ECR. So the last one in adults, you can see that in adults, compared to the controls with the empty one and also with the GFP dsRNA, again we would see that with the combined USP and ECRA, very obvious deformities were observed, and again with the ECR and USP and VAP. So what it mainly established, we noticed that yes, the dsRNA have direct lethal as well as sublethal effect. And again, you can see that we checked the effect of these dsRNA on end-plane behavior on the adults, and you can see compared to the controls in dsRNA with the USP and ECR, 87% reductions was noticed. 87% reduction in egg-laying behavior. If you look at the number of eggs in control, 136, and in dsRNA in adults feeding on the USP and ECRs, you can see the 16. So 87% reduction. This was really interesting results. And again, we were looking at the effect of these dsRNAs on egg viability and hatching rate. And you can see again interesting rates. Interesting hatching rates in control, and also in USP and ECR treated foliage where these the insects were fed. So all these results basically led us to our hypothesis that yes, the tiny dsRNA molecules can really inhibit the insect growth and can reduce their populations. So at the end, yes, by the way, we have another project by the Tugatag, and we are trying to load these dsRNA and developing into a kind of biopesticides. And what we believe that because this biopesticide is a non-GMO, eco-friendly, it will be equally effective and equally acceptable by the regulatory bodies and farmers, not only in Turkey, also in Pakistan. So I'm very grateful to the TAG for providing funds to the grants. We have another recent grants from the TAG to develop these dsRNA in the form of biopesticides. Also, we are thankful to the private group, that's the Deutsche Research and Development Group, that also funded this project, and still they are funding. And also, I'm very grateful to the Dr. Ayhan Gokce, who is the entomologist, who kept on monitoring all the entomological parameters in this study. Dr. Mehmet Chalishkan is a very renowned potato breeder, and continuously supplies us the potato. And also, we are working with him for the agronomic experiments with him in future when we will transfer these, or when we will try these biopesticides in the field. I'm very, very grateful to Dr. Emre, entomologists, bioinformaticians, who is always there who has been there all the way this project. This all research work was conducted by PhD student Dr. Mif Tariq, who is in China for his postdoc right now. And I'm grateful to all of you for listening me, and I will be really happy to take any questions. Thank you so much.
If you have any questions, we have the email of Dr. Allah Bakhsh, and maybe by the end of our session, he will provide the answer to your question. So if you have questions, please, you can give them to me. Okay, our next report is by the head of the laboratory, Belarusian State University, Mr. Maxim Potapovich, and the topic is therapeutic prophylactic preparations.
Пожалуйста. Так, мы, как договорились, в течение...
Хорошо.
Please, just about 10 minutes. Good day, everyone. I would like to greet everyone from Belarus, from Minsk City. My name is Maxim Potapovich, and I am the head of Biotechnology Laboratory. I work at the Department of Microbiology at our university. And let me tell you briefly about the results of our work. As this section is related to the agriculture, it is clear that all these works will be related to let's say, to animals, to domestic animals. As I have seen in the program, there are lots of reports on some plants. However, I will be speaking about animals who are eating these plants. So for more than 10 years in our laboratory, we have been working on the development of veterinary preparations based on recombinant proteins. And mainly I will be talking about cytokines of animals, and today I will tell you briefly about it and maybe about some other things as well. Where shall I direct the clicker? Does it work? I'm trying now, but okay, now I can see it. But it is on. However, it is not working. Can we please have the assistance? Something is wrong with the clicker. Maybe I'm putting it into the wrong direction. Or maybe the battery is off.
Возможно. О, что-то пошло. Или это оно с компьютера? С компьютера, да? Переключается. Ну, следующий давайте.
Okay, then could you please show me the next slide? Well, the disease of animals is a big problem, and in every country it brings to the large economic losses because agricultural products are the products that we live with. And as I have already mentioned, in our laboratory we have decided to try to solve this issue, and we started to prepare veterinary preparations. And as I have told it, they are based on recombinant proteins, but not just proteins, animal proteins. So we have the species specificity. For example, we have the large cattle, we have different preparations for each of the type of livestock. And these preparations contain recombinant proteins, cytokines. So they are identical to the protein that are also in the cow or in the pig. So it's not something universal, because you might also know that we have some antiviral preparations, but there are just few of them. I mean, the effective one and those who are acting against viruses. However, for animals, we almost do not have them, and in many cases, we use either some specific medication that would be used for a person, or maybe it's just a symptomatic treatment, because we do not have some antiviral substances or preparations for animals. And so we have decided to resolve this issue, and we started to develop our products, drugs. And as I have already mentioned, we have cytokines that are in our drug products. As I have already mentioned, these acting substances, recombinant proteins, they are identical to the proteins in animals. So we use we use E. coli here, we use a specific mechanism, we receive substances, we check the mechanism, and we use it as a basis for different medical products, veterinary medicine. I guess you all know what cytokines are, because this is about large group of molecules, and there are lots of proteins, and some new substances are being discovered. I think that last year Chinese people discovered the fourth type of interferons. Before we had just 3, but in 2023 there was an article about this fourth type of interferon. As you might already know, interferons, they do not directly act against the virus or bacteria or any other pathogen. Their action is the following one. They act on the cells, and they produce this specific resistance state, or resistance to the penetration of some specific viruses. So it's, let's say, not direct but indirect action. And the idea that is on the basis of our medicine is that we install cytokines in our animals, and the animals' own protective mechanisms would be initiated, and due to this, the cow or the pig or the dog, using their own immune systems, would fight against pathogens. And of course, it is clear that this is an environmentally friendly product, because such proteins, they do not have any specific, expiry date and so on. These are not microbiotics that you would receive somewhere. Sometimes you have to put some substances before you slaughter the animal or before you milk in the animal. But we know that we cannot use a product or the meat of an animal if the antibiotics was in it. But now this is environmentally friendly, and right now we have the series of veterinary medications, and as far as I know, they are sold in Kazakhstan as well. Right now our countries are in Eurasian Union, Customs Union, and so the system of medication registration is unified. So if we register it in Belarus, we have the right to sell it all over Customs Union market. We sell in Russia, we sell in Kazakhstan, no specific limitations or boundaries, and we sell it without any specific necessary permitting procedures. As I told, we have specific medicine for cows, dogs, pigs. This is what we produce, and this is what we sold. And our many years experience now turned into the product that you can touch, so the tangible product. So this is something that was produced. Next problem, and today during the plenary session, people were also noting it. So we speak about multiple antibiotic resistance, and we know that each year it is more and more difficult to, let's say, to fight with it, and there are always some new let's say, bacteria and no other antibiotics could be used, those who exist. And nowadays, there are no alternatives, because pharmaceutical companies do not want to invest in the development of antibiotics, because it's about billions of dollars, and they do not want to spend years or 10 years for developing, for trials and so on. So no antibiotics are produced now, and we use the existing one, but the existing, they are inefficient. And here we also have a specific tool, and if we if we look at the antibiotics that we have now, right now we have some specific limitations with regard to application of antibiotics. However, now the rules have been strict, but the animals would use antibiotics, then it would go to manure, and then it would go to the soil, and then it would go back to a human. I presented my report a couple of years ago in Moscow Moscow, and it was the director of one veterinary institute from Voronezh, and I was speaking about this problem, and he told me that they also had this task, and they wanted to find the soil samples for the analysis, but it should be soil, like clean soil without antibiotics residues. And he told that he was not able to find such clean soil, because there is no. Maybe you heard the story about the Thames River. The story was a rumor that people say you can even take or receive penicillin from the water of the Thames River. I mean, our soil, our water, everything is contaminated with these antibiotics, with these residues, and the more contaminated the environment, the worse effect it would give to us, because we are people and we produce products from soil, we drink water and so on. Okay, what we can do? Well, we understand how all of this is being spread out. We have the, let's say, the cycle of all these antibiotic substances and the residues all over the cycle. This is the specific research. We see the number of resistant strains is increasing every year. Why? What am I going to speak about now? We have bacteriophages, or the bacteria viruses, and they are parasites, and these phages, they have specific enzymes. And they are called lysan or lysosines. This is the enzyme that is in a very specific place, cuts the wall of bacteria, and then bacteria would be spread out. So these enzymes, they are very specific, and their specificity, I mean, they're very specific up to the type, up to the strain. For example, if we are fighting Staphylococcus, this lysin would not impact normal flora or would not impact some other infection. This would be effective only for Staphylococcus. And so the target for these lysins is a very conservative. And the relation that is shown here in yellow color during the evolution and bacteria expanding has not been changed. So this is a target, and through this specific conservative nature, there is no sustainability. We receive the tool that is strictly specific, and it doesn't initiate sustainability. So, in our laboratory, we received such proteins, and we have 2 or 3 of them, one against Staphylococcus, and the other is against Streptococcus, and the third one against Enterococcus. Your time is over, Just 2 minutes and I'm going to finish. And as I have already mentioned, if we put this protein within this protein in a specific environment with the Staphylococcus, in 5 minutes, nothing will be there. It is a specific structure of this, like this one. The structure is the same one. So it's like they are drilling the hole in the cell of the bacteria, and then the content of the bacteria would go on. And let me also tell you about the direction that we work with. There are antimicrobial peptides. Frogs, they live in a wet environment, and this is a heaven for bacteria, right? And evolutionary, they have this feature. They have the set of peptides. It's not like previous proteins that would influence one specific bacteria. These are peptides that have a general nature. They would impact all bacteria. They are positively charged. They have lots of amino acids, and so they, they do it the same way, but they also make a hole in a cell of bacteria, and bacteria would be like broken down, and the content would go out. So we did one of the reports. So this is antibacterial activity. Well, basically, this is it from my side. And if there are any questions, I don't know if moderators would let us. Any questions? Any questions to Maxim? I think that during coffee break we will ask a question to you. Thank you. Okay. Thank you. Our next reporter is Ms. Larisa Sherbakova. She is the head of laboratory, All Russian Research Institute of Phytopathology, Russian Academy of Science, Moscow. And her topic is detoxifying aflatoxin B1 and zearalenone, recombinant enzymes as innovative tools for biological contamination of cereal feed. So please. The person is not using the microphone. I want to thank organizers for invitation. As any other microorganisms, mycelial fungi produce many secondary metabolites of different nature, including mycotoxins that have toxicity to warm blood, and they produce different pathogens. They are pretty sustainable in the process of production of food and so on. For example, fusarium can produce more than 190 mycotoxins, including aspergillus can produce more than 20 different aflatoxins and their derivatives and their closely related compounds. Iambiazine, the main producer of which is Aspergillus flavus, parasiticus, and nominus. And these mycotoxins and other different aflatoxins like B, iambiazine, they are typical contaminants of feed, crops, grains, and some other ready-made feeds. The problem of mycotoxin contamination is a global one because compliance of the safety of feeds is the main factor for its demand at the global market, and the contamination with mycotoxin, including aflatoxin B1 and zearalenone, decreases the competitiveness of this product. That's why this is the problem, and problem of contamination, decontamination is of great interest. As any other mycotoxin, IFB1 and others, they are included in the food chain. And you know, the term aflatoxin appeared in 1960s when in England, it was a loss of animals, mass extermination of animals, or the animals died because of the feed that was contaminated. So aflatoxin is a strong mutagen, and it is a cancerogenic, the most cancerogenic substances. Zearalenone and its derivatives, they are pseudo-estrogens that cause hormonal imbalances and infertility. And you know, the problem is the following one. Aspergillus and Fusarium, they are parasites. And they have a specific lifestyle, and they would populate all substrates, and contamination could be performed during harvesting or some other agricultural procedures. So that's why it is difficult actually to prevent infection, and so that's why even fungicides, antagonists, and resistant varieties, they are not always efficient. We have different biological and non-biological methods of decontamination. Here you can see advantages and disadvantages. All of them have their own advantages and disadvantages. And out of biological methods, the most environmentally friendly and the most attractive now is the approach that is connected with enzymatic decontamination. So the use of microbial enzymes that attack structure of mycotoxins, and by this they lead to the formation of non-toxic or less toxic derivatives. So this approach has a number of advantages, and it allows to provide the safety of the feed without damaging its nutritional value. And not always we can have it when we use some other methods of decontamination, like mechanical or physical one. Many microorganisms, but not only microorganisms, for example, here we have the fungus, bacteria, they are sources of such enzymes. And as enzymatic decontamination is of great interest now, and now they are studied, they are being identified, their structure, but in fact,
таких ферментов, для которых была бы подтверждена безопасность продуктов деградации афлатоксина, не так много.
There are not so many enzymes that would be safe. And here we want to speak about AFB1 oxidase. It is a specific one, and we also have Calahakis rosea. So these enzymes, they were the subjects of our researches. And you know, the thing is that they are high efficient decontaminants, but absence of the available technology of receiving of some, I mean, there are some technological challenges nowadays. I'm not going to dwell on it. However, these challenges could be overcome, and we could do it in easily cultivated and high productive producent, and this approach allows us to provide feed safety and to preserve nutritional value. Besides, it is very comfortable for feed treatment, and so our task was to receive this specific substance that would have all necessary property and that would be used for decontamination of grain or wheat. Besides all these properties, we had some specific prerequisites, and I will not speak about them so that to save the time. Here you can see they are specified for these elements, but these are different products. And oxidase was in yeast cells, and hydrolase was found in some other. This is eukaryotic ecosystem. This slide and the next slide will show you the protocol of expression of aflatoxin oxidase and the second element. They include all necessary stages of receiving recombinant proteins. So we have the specific vector mechanism, and I want to say that when we speak about aflatoxin oxidase and when we speak about specificity of our system, we include gene of aflatoxin oxidase to a specific procedure, and then when we use зеленую гидролазу, we used additional plasmid, and we used highly efficient xelanase promoter.
После того, как наличие, после того, как методами прямого секвенирования и
using the direct sequencing method and PCR, we proved the correctness of the rate. Then, using the specific method of electrophoresis, we identified the absence of the target enzymes, and we also did the selection of most productive clones. So, using the specific procedure, we received the preparations, and then that were lyophilized. And then, before testing them on the contaminated grain, we tested it in vitro, so that to choose best conditions, so that to check it. And so here you can see that in the optimal conditions that are specified, we can reduce up to 80% of aflatoxin from the model solution, and zerno hydrolase in 4 hours would totally reduce this aflatoxin. As for modern rules, the EU countries have a specific permissible limit of AFB1 for feed grain, 5 microgram per kilo, and the US grain safety rules allow 20 microgram per kilo. However, when we have samples, we see that the level of contamination could be really high. So in our experiments, when we started to test recombinant proteins at contaminated by aflatoxin grain, and it was made by injecting the high toxigenic strain. So we received a grain of high level of contamination. And although we had so you see here the low numbers, the numbers at the basis is what we had. You see that residue content is still higher than permissible limit. However, if it was the average or higher average level of contamination, we would have the other numbers. So you now can imagine that if it would be not so high contaminated samples, we would receive a better effect. As for the other substance, in 12 hours, the percent of degradation. In case of wheat, it was 63, and in case of corn, it was 53. In a day, we had from 2 to 9% in the wheat and corn. However, in 48 hours, the grain was fully clean from toxin, and the chromatography didn't identify any contaminants. So we were able to receive quite efficient enzymes that could be used as the potential or as a basis for elaborating decontaminant products. Of course, there are some, let's say, limitations, but we are still doing some additional research so that to eliminate all these limitations over the years. So we want to prolong the stability, and we want to receive a mutant form of
хорошо работает при
the
более
ZEA.
кислых pH, чем, потому что в кормах, когда
You
мы
know,
привозим
in
с
some
молоком, этот
feeds,
pH
when the
8,5
feeds
это
are
достаточно
produced, they
непростительно.
have the specific
Так что первый результат
level.
уже
So
есть
we
успешный.
do already have first
Да, в
successful
заключение я
result.
только
And in
хотела бы
conclusion, I
сказать, что
just wanted to
по
say
существующей
that
концепции
according to the
внедрение
existing concept,
деконтаминационных ферментных
the
препаратов
implementation
это
of
дело времени, и наряду с другими
decontaminant
исследователями
enzyme
наша работа тоже вносит вклад в продвижение
products
этого.
would be very popular, and we are promoting
Спасибо
this
за
approach.
внимание.
Thank you. Thank you so much. Any questions? Do we have any questions? Okay, maybe during a cup of tea, people might ask some questions. No questions. Okay. Now we would like to give the floor to the head of the Department of Biotechnology and Microbiology, Gumilev Eurasian National University, Professor Masalimov Jaksalak. Please. Let me greet all of the participants of our conference. And the topic of my report is the impact of combined stress on multisystemic plant defense. As you all know, stress is a specific state of the organism that could be received when there are unfavorable conditions or maybe some physiological conditions of specific nature. The plants, if we are going to speak about plants, and plants are the organisms that cannot avoid stresses. They cannot act like animals or like people to move away from the stress or to be protected from the stress. They live in the conditions where they are. Types of stress that might influence plants could be divided into several groups: abiotic and biotic. Abiotic stress, any factors connected with the nature, some drought, temperature deviations, maybe excess or deficient of light, salt stress, ultraviolet. Or biotic stresses, some factors from the life, nature, bacteria, viruses, fungi, insects, pests. Recently, there is a great interest among researchers to the stress, and we are speaking now more about combined stress. So, for example, if we take the real situation, usually there could not be one stress. It is usually the combination of different types of stresses, right? And here on the photo here, Trends in Plant Science. This is a journal, and they showed here some types of combined stress. So in some cases there is a negative stress, you see here. And basically all the work that I'm going to tell you about is actually the work conducted by two subgroups of our laboratory, and our two groups works with this topic, stress, and some other related issues. As you know, any oxidative stress connected with the different types of oxygen formation. And in our work, we use temperature stress, and we combined it either with a drought or with biotic stress. And as a biotic stress, we used the influence of viruses to the plant. This drawing shows the work, but this work was done long ago. And here you can see the mechanism of formation of the active forms of oxygen under the heat. And so we used different types of water and solutions. Usually these are solutions that are closer to the blood serum. We used salt water or seawater. And here we showed that the temperature as a constant factor of ambient environment might affect or influence influence the conditions. And as I have said before, oxidative stress used different parts. So here we use different elements like hydrogen peroxide and so on. And so we see here the formation of some damages of our biological micromolecules, such as lipids, proteins, and DNA. As you know about lipids, here we speak about different oxidation of lipids, and there are also some other number of products, proteins, and oxidation, DNA, oxidation DNA. We have there are lots of substance, lots of other biomarkers. As you know, multi-system plant defense has several systems of defenses. First of all, antioxidant defense. We have fermentive and non-fermentive part of it. Then signaling pathways, change in metabolism, activation of resistance gene. I forgot to add here we speak about reparation mechanism. As I have mentioned here on this slide, you see the research of one of our subgroups. So we did the research on the barley, and here we combined the influence of high and low temperature, high-low temperature combined with drought. And we found out that the main effect here is due to the temperature. However, the cumulative effect was not a big one. So I mean the main effect, 80%, even within this combination, not the drought, but the temperature influenced the situation a lot. We looked here and analyzed, examined some morphological properties, level of chlorophyll. We also looked at the content of the main active forms of oxygen, like superoxide radicals and hydrogen peroxide, and we found out that the high temperature influences a lot, not the low temperature. However, when we speak about roots of the plant, the process could be vice versa. But that's the main effect was found here at the high temperature, and actually it is correlated with our previous researches. The growth of active form of oxygen influence, even if it's 40 degrees by Celsius for barley, it is not so, let's say, it was not so effective. However, we tried also to use the right level of humidity, not more than 50. We have also examined the system of antioxidant enzymes. So you see here, we did the analysis. Mainly you used the leaves of the plants and roots of the plant. And again, the temperature is high, and this high temperature gives a high effect rather than the drought. However, if we did our research maybe for a longer period of time, so maybe, yes, the drought would also, we would also find out the importance of drought in all this process. We also looked at the proteins, HSE90, HSP70. Again, we can receive interesting effect when we observe high temperatures. Apart from this, we have also conducted the research on combined effect. We used again high temperature, low temperature, plus we were also using the infecting with a virus. We used Nicotiana bentamiana plant. We did also the research on some morphometric indicators. We checked the content of superoxide and hydrogen peroxide. We also looked at the antioxidative defense. We looked at the leaves. We looked at the roots. And in each of the case, we see the strange, let's say, influence. Why we started these experiments? We were really interested in some of the facts. One of the strategy how plant fights against some viruses is through the formation of the specific forms of oxygen. So first we did the stress with the temperature, and then we kind of, let's say, vaccinated the plant, and then we tried to check if the plant would survive. And you know, these plants, they actually were more sustainable. Besides this, well, actually I want to say that some of our works, they are published, some are still under progress, we are still working on some things. We were also studying the influence, the specific influence of glycosylates. We found the analogy. Also, right now we are trying to examine the combined effect of combined stress for some other substances. Besides, I mean, here it is written that we do evaluation of the activity level of DNA repair enzymes. However, now we are working with one of the new proteins that was discovered just recently, and we are doing the research, and actually the number of research with regard to this is really increasing. This protein is pretty interesting, and we see here the suppression of key RNA, short RNA biosynthesis. We have also received pretty interesting data with regard to effect of temperature to CRISPR-Cas13 system of protection in plants. But we haven't published this data yet, and we are still elaborating or finalizing the work. And you know, actually, the temperature influences a lot CRISPR plant 13. So this is a system against RNA viruses. Here you can see we listed the people of 2 our groups who did the research. Okay, thank you so much, Mr. Jaksylyk. Any questions? No questions. During coffee break. People say that during coffee break they are going to ask more questions. Okay. Our next report will be presented by Mr. Alexey Volkov, who is the head of the project department in PREN LLC, and the topic is organization of planting material production using microclonal plant propagation methods. So please, good day, dear colleagues. Today I would like to talk a bit about the production of the planting material and about the problems, and mainly here we speak about industrial production of plants in vitro. As we all know, in laboratory, I mean especially those who are dealing with microclonal activities, you all know that in laboratories, even if we do not have any or really clean conditions or sterility, you can receive in vitro plant, and you can go all the stage from one stage to the other without any sophisticated technologies. However, when we speak about large scale production. And when we speak about any technologies, we have some risks, and of course we need to mitigate such risks. And for this, there are some specific standard operational procedures and rules and guidelines that help us to avoid mistakes. And of course, there are some other technical means that allow us to ensure the quality of any industrial technology and to receive positive results. The biotechnology itself has a high potential because many agricultural plants, when we speak about selection, breeding, new types, I mean, everything is connected now with biotechnology. The problems of agricultural industry is the following one. Many types of plants, they are brought from abroad, and there is a company monopoly that deals with one specific plant variety or one specific type of plant, and then the products would go globally. What is the problem? The problem is that we see losses of planting material during transportation. Plus, we have risks that we can, jointly with planting material, some new bacteria not let's say, relevant to this region would appear in the soil, would appear in some other plants, and as the practice show, up to 50% of planting materials that is brought from other regions either would die or has some other disadvantages that would lead to the loss of the products or to the loss of the plant itself. And now many companies, to avoid this, they try to produce their own planting material, or the laboratories that produce planting material in the region, and then this laboratory would disseminate it among its facilities. It allows to receive the district-specific plant variety, and within this activity it would be a specific work ensuring that there are no viruses. It also allows to create a specific database in the form of a gene bank, and this plant could be used in the industrial agriculture. Biotechnologies are used in agricultural industry, in forestry, in pharmaceutical industry, and of course they're used in a large scale. However, at any type of work, as I have mentioned, I'm pretty sure you all have mothers, grandmothers, and your mothers or grandmothers started to preserve or to make some preserved jam, and she can make 5-10 jars with a jam. But if you ask your grandmother to make 1,000 jam jars, then of course her very delicious jam will turn into something bad. It might start having fungi and some other. And this is related to the mass production. The same happening in the laboratory. The absence of some specific barriers or the damage of technology brings to the infecting your product with a fungi. And later I will tell you the economic consequences of it, and you will see amount of money that or the losses that people might incur. So fungi and bacteria, they are the main causes. They are in a specific environment, and they would be in laboratory. They might come through some ventilation system, and actually it's quite difficult to, let's say, to remove them. You might have it in some equipment and in some other systems, and you have to clean it too. So again, here we speak about the losses of planting materials. Up to 50% of planting materials, according are lost at the process of adaptation. Unfortunately, the process of transporting plants from in vitro to the natural soil, to the natural location, actually this is due to the weak development of root system, and they have some weak features, and for 2 or 3 weeks we need to ensure the right parameters in which this plant would be located. And then this helps plants to resist these unfavorable conditions gradually. We know that there is a specific water transpiration, and when we speak about industrial scale, many plants would die, and this is actually the main reason why we have such problems. But we have solutions to fight with this problem, and later I will tell you. First of all, we need to have the right arrangement or layout of our laboratory. It is the same like pharmaceutical industry. Here you have to monitor all types of production, starting from the beginning up to the final output, when from your laboratory this material would go to some other sites. So that to receive a qualitative material, you need to make it healthy, and there are different ways. You can use cryotherapy to recover your plants, cryotherapy, and some other therapy our previous colleague mentioned. So there are some specific cameras. The temperature is 38-40 degrees, and you receive a healthy part of plant that for 90-95% would be free from any pathogen. It is the same like for people. We increase temperature and protein compounds cannot be developed, so there is no synthesis, and you see that this is a plant, and then you use it and you work with it in laboratory, so that not to have any unnecessary materials like fungi or like bacteria. You would do all of this in clean facilities of laboratory, where you have corridors, where you have some specific parts or sections where people would change their clothes, so where there is a sterilization part, working with specific boxes, and of course you need to have the collection of your healthy materials. There are some light facilities where the cultivation of your plants would be going on, and the specific facility for adaptation of your material. As for adaptation, we can use different types. It could be some cameras, it could be greenhouses, but in rare cases. Why greenhouses? Not good to use greenhouses. First of all, it is difficult to provide relevant microclimate, because anyway the natural conditions like the sunrise and sunset would impact the temperature. And again, we speak about the surrounding nature, biofactor, I mean the spores or the fungi could anyway could be found out within the plants. So again, when you produce planting material in laboratory, I kindly ask you to pay attention to the correct layout of the facilities in laboratory. You need to select correct equipment, because all of this is about money. If we for example, let us say we have laboratory with the production of 1 million plants per year, and here you see that from 0.3, 0.7 euro per unit, these are the costs for your plant in vitro. And each 5% of your plants would be lost, and this is 25,000 euros. And again, if we take the whole cycle, this amount of losses would be even higher. So if you have a mistake at the very first stage when you organize laboratory or when you use wrongly selected equipment, would give you later such circumstances as a money loss or financial losses. If there are any questions, I am ready to answer any questions. What types of plants you are cloning? Any. Do you have some herb? Well, yes, we work with different types of plants. We work with some pine trees, and we can also work with pine trees. We use different biotechnology, but biotechnology is used mainly for agricultural products or for some breeding activities. Okay? Thank you. Any questions? Okay, then according to the schedule, now we are ready to announce coffee break for 30 minutes. It will be here on the first floor. And in 30 minutes we wait you back here.
Скажите мне, дайте гарантию. Больницу там зал большой, знаешь, кинотеатр. И у них стулья были укреплены. Там было жутко холодно. Меня накинуло какую-то тулю. Приехали нас возили, там было еще советское время. Вот. И я вошла, значит, между рядами пошла, и этим пледом задела эти ряды где-то на уровне 10-го ряда. Он такой идет на солнце. И они стали подскакивать спинку, и они стали складываться. Ложиться, они изумлены, какой там цветок. А как-то прославилась.
too bright for you?
But I anyway have to stand there because of the microphone.
Yeah, but they can change the lights. I can see because the lights are bright. Would we
Yeah, it's quite bright. Yeah.
Bunlar var mı ki ne yerike? Kötü metalmatım buna kın. Виталий Георгиевич. Виталий Георгиевич.
Дальше у нас Павлина. Видно.
Павлина, да? Есть?
Есть. Дальше Степанюк Наталья Владимировна.
Пожалуйста.
Спасибо.
И последний.
Извините, не могу потеряться. Я практически все заканчиваю. Я очень организовала классе, на родине.
Я хотел спросить, у вас вот рекомендантные есть, да, вот это первый?
Ну, есть, да, есть. Но мы работали, но, правда, сейчас всех их израсходовали, да.
Так всегда, для ремонта. Тоже кишечная пауза.
Нет, в смысле, мы сделали не в Шерихе. Мы сначала делали, в смысле, у нас были экспрессии в этой самой прокариотах, в Шерихе были. Но у нас идея как раз вот была в эвкалиптических системах, то есть дрожжевых клетках и в этом пенициллиум канестес. Он чем интересен, это вот наши коллеги из Лимаха, штамм, который имеет очень развитую систему целлюлазных и ксилоназных ферментов. И его можно использовать для повышения питательной ценности кормов. Он разлагает плохо разлагаемые полимерные углеводы. И вот мы туда добавили, значит, как бы эту зеленую гидролазу, то есть он как бы повышает и питательную ценность, да, может, потенциально, да, и безопасность. Вот. И еще есть идея туда вот оксидазу вставить, но там сложность в том, что у них разные оптимальные pH сильно различаются, вот, поэтому вопрос. Но сейчас вот мы пытаемся сдвинуть как раз вот зеленую гидролазу, сделали у нас в Баке, мы с ними работаем, они сделали несколько мутантных форм, гидролазы, с тем, чтобы сдвинуть оптимум pH, да, вот. Она практически еще работает, а вот репетирует совсем.
Tu vous avez свои или конокерика? Или конокерика или...
По-моему, нет. Аска не работает. Не у меня, во всяком случае. Ну, огонек не видно.
Акса могла быть ничего. А вы после этого в Караганду-то приезжали?
Нет, я после университета я уехала на
Так, по программе мы должны уже работать.
So, according to our program, we need to start our work. So, our first report after the coffee break is presented by the head of the laboratory, All Russian Research Institute of Phytopathology, Russian Academy of Science, Mr. Vitaly Javahia. And his report is on the topic study of the molecular mechanism of the protective action of the MF3 protein from Pseudomonas fluorescens, which induces plant resistance to diseases. Please, Mr. Vitaly, the floor is yours. As far as I understand, I have 15 minutes. I have 15 minutes, right? Yes, you do. You do have. Well, first of all, I would like to express the gratitude, and actually previously Miss Larisa Sherbakova expressed the gratitude on behalf of the facility, but let us again also express the gratitude. Today I will be talking about two types of protein that are able to induce plant resistance to pathogens. No, I have it. I have it. The modern agriculture cannot be without synthetic pesticides that protect our harvests from pests, from insects. However, at the same time, being xenobiotics, the synthetic pesticide contaminate soil, water products, and other things. So right now we see the tendency in finding alternative ways or to use the molecules of biogene origin that would not have such, let's say, strong biological consequences. And among such specific products or substances, we are speaking about proteins because they are safe compounds that would leave behind natural amino acids, and it is quite easy to work with them. We can manipulate different structures, we can study, and we can increase the optimal activities. But I will speak about two proteins. Well, first of all, one of the proteins that we give a name to him, ML2, it is cold shock protein, low molecular, thermostable. It was isolated by us from Bacillus thuringiensis, and the other low molecular thermostable protein, which we called MF3. This is a microbial factor 3. MF3 was isolated from Rhizobacterium Pseudomonas fluorescens. Such proteins are able to induce resistance properties. And you know, the structure of these two proteins actually is different. So these two proteins, they differ a lot. Here the slide shows the ability of these proteins. Here you can see to which pathogen this protein, with regard to which groups of pathogen this protein works. So here you can see it. You know what we did? We did some specific experience, and we worked with different pathogens, and actually we found out that the mechanism of these proteins are the following. They affect or influence the disease through the plant. As the proteins had different structures, we supposed that the activity mechanisms are also different. And if it is this way, then I think if we combine them, we would find synergies or synergy. We suggested that these two proteins would affect different receptors, and if we combine these two different activation factors, probably we would receive a higher effect rather than to use these proteins separately. And actually, it was in fact this way. The combination of these proteins worked better than these proteins in separation. So we decided to use hemeric protein from these two proteins. And here you can see the structure. You know, we received the grant from RNF, and jointly with the Center of Bioengineering or Center of Biochemistry, we I don't know, it's a new name, but actually it's a former Institute of Biochemistry. But we work with them together. So here these two domains were united. The hybrid protein was created as a structure consisting of two domains. And we received the data from electric phonogrammy, and we understood that this protein and its molecular mass is complies with the hybrid one. So you see how everything here looks. Then we transformed one of the strains of E. coli. It became the superproducent of this hybrid chimeric protein, and it was shown that this hybrid was also better and more active rather than the different separately, and the activity is the same. But technologically, in our conclusions, rather than working with these proteins separately, we need to take chimeric protein, and then based on this we would create plant protecting products. So we compared the combination and the hybrid, and you see, according to this slide, that they act similarly. So in this work, a chimeric protein elicitor was used for sustainable resistance induction plants. So the use of this chimeric protein resulted in reliable increase in protective activity compared to individual proteins. A similar effect is characteristic of mixtures of elicitors with different mechanisms, which further confirms our indirect data. And the results also demonstrated comparable effectiveness of the protective action of this, and we can use it in practical parts. Now I speak about theoretical part of my work, but then I also will speak about some practical things. The one and the other protein, we took them and we defined the active center. For example, as for MF3, we predicted it from theoretical point of view, but then we synthesized the 29 oligopeptide, and it turned out that we received the concentration which was pretty active, the same way as the original initial protein. And as this small fragment, actually this is that part that was an active one, and the main idea is that we start to think with what this protein interact, in the plant. So probably through this active center there is such interaction, and then we decided to do the specific structure of protein. I will show you. We see yellow color, and this highlighted yellow color is an active center that consists of these amino acids. So we've decided to find the alanine scanning when we change the parts to alanine. And so by this we were able to receive the set of mutants generated by alanine scanning. And so we tried to do it and to check. When we have received all possible options or variants of such mutants, we found out that part of the mutants, they lost their activity they quite lost their activity. So again, these amino acids, they play important role in interaction with receptors. Some part was quite high, some part was quite neutral. And based on this, we were able to do some other further conclusions. So this is the way we did it. So here we see the tobacco leaves. They were treated on the left side with MF3, which was original, and on the left, this treatment with mutants. Then we inoculated it with a virus of tobacco mosaic, and here we see that the one on the right loses its activity, and probably this amino acid plays its role in interaction with the plant. And our people of theory, they made such models. They used computer programs, and the computer programs allowed them to present it. So white color, these are those amino acids that play a very important functional role. Blue color, less activity, yellow even less, and in general we received the structure of this protein molecule. And this what we did from the theoretical point of view. Thus, testing of the resistance-inducing activity of mutants using the tobacco mosaic virus pathosystem showed that some of them decreased 2 times or 3 times the inducing activity, and these mutations demonstrated a close dependence of the inducing activity of the protein on the structure of its active center, which probably contributes to the recognition of MF3 by the receptors of the host plant. The theoretical part has been finished, although I can talk even more because now we look at the genes. You know, there are some genes that participate in protective reactions, and now we look at the expression. We look how MF3 treatment impacts some specific genes. But maybe next time we will tell it in a year. You know, people asked me, can we spray plants with it? Actually, we need to think here about ultraviolet rays and some plant proteases because they activate the protein. So later we, with the other people from other institute, made microparticle. Here we have the specific polymers that, biodegradable polymers that were loaded with MF3. And these are the data that we received, and they tell us that these particles do not have higher activity. Now we are conducting the field trials in 3 different regions with different soils, and we do the trials on potato. So here on the left you see control group, and on the right you see plants that are treated with MF3. And you see MF3 part is already blooming, right? So it means that treatment of potato with this product or preparation accelerates this flowering phase. So we also wanted to check the toxicity at animals, and we did this acute toxicity testing of MF3 on aquatic crustaceans and green algae, and it showed no toxicity. So, and it suggested the protein is environmentally safe and could be used. Thank you. I think I did it right in time. Thank you so much, Miss Vitali. We have time. So the fusion protein was more efficient than combination? No, actually there were no very specific difference. But technologically it's better to take one and to work with it. So it is like universal, right? Maybe it sounds like unbelievable, but everything that we checked and tested, you know, my students also worked with MF3, and they did do treatment of different berries, strawberries, grapes, some other flowers, the orchid flowers, and for the moment it works. Is it a... Did you use E. coli? We did E. coli, we put plasmid, we produced. And our colleagues, they also did the yeast producent. You know, actually both proteins helps in treatment. What about the commercialization process? Speaking about commercialization, what I showed you, these are industrial planting sites, and there are 3 different zones. Actually, the preparation form for agriculture is a separate issue. We did the simplest form, and according to the data, the data that we received told us that the concentration in potato decreases the X virus. What we do now, this works. And I hope that when specialists come and they would start optimizing the process, they will achieve maybe better results. They do different activity. Thank you. Thank you so much. Any other questions? Is it economically efficient? What if it is too expensive? Did anyone calculate the efficiency, economic efficiency, if we speak about industrial supply? You know, in our laboratory we have agricultural specialists, and they were taught how to calculate all of this. However, I am more interested in the mechanism of activity. As for some practical part, I can tell you the following. These field trials, they are very expensive. These are private holdings and companies. They gave us the land plot, they treated it. Now they gave an order for a bigger amount of protein, and we don't have enough capacities for the moment. But they got interest in it, and I think that they are able to count. They spend their money. So hope. Thank you. Any other questions? No? Enough? Thank you. Thank you so much, Mr. Vitaly. Okay. Our next presentation on the company's products, PCR amplifiers and centrifuges. Ms. Pavlina Müller-Kropa, Product Solutions Specialist, Eppendorf Group.
Good afternoon, ladies and gentlemen. My name is Paulina Müller, and I'm a product and lab support specialist at Eppendorf. I'm very glad to be here today, and special thanks to the organizers and to our distributor for Kazakhstan, Welt. Today I will talk about our centrifuges and our PCR cyclers. And you will hear the latest news from these 2 product groups. First of all, let's have a look at the centrifugation history at Eppendorf. This year is a special year for us. We are celebrating 60 years from the manufacturing of the first benchtop laboratory centrifuge. Nowadays, they look much different than they did in the past, but they still do their job. So basically, they separate particles based on their size. So the development through the past 60 years brought to this colorful and versatile portfolio. So from the very small and tiny centrifuges with only 12 places for the smallest 1.5 or 2 milliliter tubes, to the big and very fast ultra and high-speed centrifuges that have been recently added to our portfolio. And of course, everything in between. So there is almost no separation task an Eppendorf centrifuge cannot do. How does the portfolio look today? So here you can see all of the centrifuges, and today I will dig into ultracentrifugation world a little bit deeper. It's not moving. Oh, okay. So, how do the centrifuges differ among each other? Basically, either in terms of capacity of volume or in terms of the maximum speed or the g-force they can reach. When we are speaking about multipurpose centrifuges, the volumes are somewhere up to 2 to 3 liters, and the speed goes up to 50,000 G for the smallest tubes. But this is sometimes not enough. Yeah? Certain molecules, very small molecules, viral particles, extracellular vesicles require higher speeds. And this is why we have added to our portfolio also the ultracentrifuges that can reach up to 1 million G force. And these ultracentrifuges, of course, as you are used to in Eppendorf products, also have some special features, unique features that can facilitate your everyday work in the lab. One very important feature in ultracentrifuges is the automatic rotor life management system. This is a system that will actually enable you to know when to retire your rotor, not based on the number of runs you perform, but based on the actual force that has been applied to the rotor. Because you know that ultracentrifuge rotors work in very, very harsh conditions. Usually the chambers are refrigerated, and the force that is being applied onto these rotors is enormous. Yeah, we are speaking here about hundreds of thousands of G-force. And this is why at a certain point the material is so worn out that the rotor needs to be replaced. But when does this point come? Most of the producers of ultracentrifuges out in the market simply count the runs. They tell you, okay, you can use this rotor for 20,000 runs, and that's it. Not taking into consideration how these runs are. Are they short? Are they maybe done at a very low speed? Because you know that a short run at low speed will not influence the material of the rotor as much as a very long run at maybe 600,000 G. This is not comparable. And this is why the automatic rotor life management system actually does not count each and every run as one run, but it can only count half the run if you did it only for half an hour, or if you only used the half of the maximum speed the rotor can go. And when you get the message from the centrifuge that this rotor should slowly be retired, you know that this is not the administrational end, but it's really the physical end of life, because the metal has endured and suffered so much force that it's destroyed to a point that it's not safe anymore. And this system increases the rotor life significantly, because rarely you use the rotors all the time at high speeds or at maximum speeds. Second very nice feature, I will show also imbalance, vibrations, and the centrifuge will stop. In ultracentrifuges, this is even more important because the forces are so much higher. And you would have to balance the the samples within a tenth of or a hundredth of a gram. And this would be very tiresome, like in this situation. So you are loading a sample under your hood, and then you bring it to the scale. You realize that the two samples are not balanced. Then you have to go back to your hood, again maybe add or take away some of the liquid, and go back to the scale to check the weight again. And you could have done it maybe 3, 4, 5 times, depends on your skill or usually just on luck. What Eppendorf ultracentrifuges enable you is just to visually balance the level of the liquid in the tube within a few millimeters. So what you can see with bare eyes, if the levels in the two tubes are more or less the same or within a few millimeters, that it's okay. You will not get an imbalance. The centrifuge will feel no vibrations because the attachment of the rotor and send out these oscillations and vibrations, and you will have a very steady run with a very easy filling of samples. Okay, I hope ultracentrifuges have amazed you, and now it's time to speak also about Eppendorf master cyclers or PCR amplificators. Recently we have released the new master cycler X40, which has a familiar but refreshed design. It is very reliable and produces reproducible results for very variable volumes from 5 to 100 microliters per well. And you can also use single-dimensional gradient in these cyclers. Also, you know that Eppendorf pays quite some attention to ergonomics. So the safe lead, regardless of how you close the cycler, it will always apply the optimum pressure to the tubes to avoid evaporation. And you can do it always with one hand. So in the other hand, you can hold the plate or whatever you still need in the lab. The interface is very intuitive. If you are able to use one of Eppendorf devices or one of Eppendorf cyclers, you will be very familiar also with this interface, so no additional studying of the manual is needed. The device has become a little bit smaller, which is also important for your space in the lab. And the cyclers can also be connected to one another. In addition to X40, there's also, I would say, the state-of-the-art master cycler X50. This is a more advanced model because it offers in a silver block the ramping or the heating speed of 10 degrees per second, which is very important for clear results. You can have 2D gradient, about which I'm going to talk in a second. You can connect up to 10 or up to 50 units together. The lid, it's also a bit special because it applies slight vacuum and very even pressure to the samples, so you will avoid any evaporation, and it's whisper quiet. Okay, cyclers are sometimes very noisy in the lab, and that's a bit uncomfortable. This one is really quiet. Can we start the movie maybe? Let's see if it works. No? No.
Тут опять не заработал. Посмотрите сюда.
Нет, там просто видео не запускается.
There should be a play button if in the lower left corner if you move the mouse over it.
Ага.
Okay. Now let me share a few details about the features of the Master Cycler X50. It comes in a few block variants, either aluminum, silver, or 384 well. Of course, silver has its advantages because of the thermodynamics of this metal. It is much faster in cooling and heating rates, and this can give you much better yield and much preciser results. I already mentioned the two-dimensional gradient. Why is it used for? By using the gradient in one dimension, you can optimize the temperature of a certain PCR step in the whole protocol. But if you use the two-dimensional gradient, you can optimize two temperatures at the same time. So you can find out what is your optimum denaturation and annealing temperature in only one cycle, which means a lot less of reagents wasted only to optimize the temperature and much faster results. And once the temperatures have been optimized, you can go for faster protocols because you know that your enzyme is working at the most optimal temperature. You can go for lower reaction volume with less template, and in the end you will get more DNA for your downstream applications, and you will also get sufficient yield in very difficult applications. Maybe also a thing for the ones who publish a lot, you will also get much nicer bands. And it's a special pleasure to have a nice photo of very clear bands in your article. So when you have a challenge in PCR, Eppendorf has a solution for you. Either it's fast ramping in cooling and heating, two-dimensional gradient, of course linear ramping, and we have center fixed Peltier elements. Up to 6 Peltier elements are present in certain devices that enable you really to have really nice distribution of multiplication. So, thank you very much for your attention.
Thank you very much, Paulina, for your information about your company's product. Are there any questions to Paulina?
I have one question.
Sure.
You said that you have 2D gradient, right?
Yeah.
And can we optimize all the places so that, for example, from the first row and first column there will be one temperature, and on the second row and second column there is going to be another temperature? How much the temperature is gonna vary between these two places?
Yeah, it depends on your setting. So when programming the gradient, you have two options. Either you set one central temperature, which you suspect is going to be the optimum, and the cycler will distribute away from it, so to a bit colder and to a bit hotter. This is one option. And if you take the Gauss curve into consideration, so the temperatures around the one you suspect to be the optimal are going to be more dense, so more closely together, and towards the two extremes they will be a bit more far one from another. Or you have the other way of setting the gradient. You can choose the lowest and the highest temperature, and you will have an even distribution of the temperatures in between. Of course, depending, the difference between the two temperatures depends on how big is the difference between the minimum and the maximum temperature.
And is it possible to program? The first place is going to be 50 degree, and the second one, the closest, is it possible to make it 60 or 80 degree
No, they are not freely programmable because usually it's the difference between the two temperatures is maybe 1 or 2 degrees only because you suspect for, I don't know, the naturation to be somewhere between 94 and 96. So you would put the lowest on 94, the highest on 96, and the device will distribute the 12 temperatures between the two. Or you put, I don't know, 94.5 in the middle, and you will see the temperatures from 93.7 to maybe 95.
Thank you.
Yeah.
So, questions, please.
Yeah.
What is your company doing on a larger scale? Is it still competitive compared to other companies?
Yeah, I suppose so. We do have.
Is it your company is growing, stable, or diminishing?
I cannot talk about financial results right here, but it depends also, of course, on the region, on the product
There are
group.
many companies on the market, so why we should select Eppendorf? Because it's often very expensive one.
Yeah, I think we have something to offer, which is, I would say, uncomparable quality. And I think, but many other companies do have that too. But I think after sales support, it's impeccable in Eppendorf. So once you buy our device, you always have someone or from Eppendorf or from our partners on your side. Yeah, I think it's not only the product you get, but usually these products are being used through decades, and it's important to have the spare parts available, to have the support and service available as well.
Okay, questions?
Thank you for your presentation. I want to ask about the ultracentrifugation. You said that that's enough to balance the tubes, like visually, it's okay.
Yeah.
And what to do if you have, for example, two liquids with different densities? I think, so, for example, glycerine in one tube and water in another. It will be the huge difference in mass.
Yeah, exactly. This was also listed on the slide. I didn't specifically stress it. This, of course, applies only to the samples of the identical liquid. This is a prerequisite, yeah? The same tube, the same liquid, because there is a certain amount of mass and vibrations the centrifuge can overcome. But yeah, when we are speaking about the density, big differences in densities, then this is not applicable. And also in ultracentrifugation, when you have samples that are denser than 1.2 grams per milliliter, you cannot use the maximum speed of the rotor, so also the speeds needs to be adapted. Okay. If there are more questions, please feel free to come to the Welt
Paulina,
booth.
does your company has a representative office in Astana?
No.
No.
We do not have.
In Kazakhstan?
No. We have a distributor based in Almaty. Anton is here with us today. So if you need any information on Eppendorf devices, please refer to Welt's booth. So, and usually the questions they are not able to answer end up with me. So now you know the face.
Thank you.
Now we would like to give the floor to Miss Statsuk Natalia. She is the head of the State Collection of Phytopathogenic Microorganisms, All Russian Research Institute of Phytopathology, Russian Academy of Science, promising biodegraders of polyketide mycotoxins contaminating agricultural products. Please, Miss Natalia, the floor is yours. Good day, dear colleagues. In my report, again, we will talk about ecotoxins or mycotoxins that Ms. Sherbakova started. Actually, we should have the different order because now I'm going to describe the previous stages. But okay, what we have, we have. One short slide. I just want to present our institute. In 4 years, we will celebrate 70th anniversary, and in Russia, it is the only institute that is focused on phytopathology. I mean, as a whole organization, it's the only facility on phytopathology. And here you can see the number of our researchers, employees, departments, laboratories. You know, when we had joint grants with the US, Canada, and some other countries, we received modernized equipment. And actually, we have all the necessary equipment for conducting researches at all levels, starting from laboratory researches, greenhouses, climatic cameras, and trial fields. And also, separately, let me mention that within our institute, we have the state collection of phytopathogenic organisms. Well, actually, my topic and topic of Ms. Sherbakova would pretty have some common ideas, so I will not speak in more details about some of the slides, but let me tell that aflatoxin and zearalenone are polyketide secondary metabolites, and they have a number of negative consequences. Aflatoxin B1 is a carcinogen, zearalenone gives let's say unpleasant estrogenic and anabolic properties. It makes the disorders to the reproductive system. I have more recent data than Ms. Larissa gave us. I want to say that contamination of agricultural products with AFB1 is the problem of southern regions. However, the EN is in the zones with moderate climates. Still, we see a quite high level of contamination. If we speak about Eastern Europe, including Russia, then contamination of zearalenone would be in half of the samples in the organization that does such analysis. As for decontamination, again here briefly, I want to say that physical or chemical decontamination is is the only variant. It contaminates the environment, it worsens the quality of products. Mechanical sorting, washing, husking, all of this allows to reduce the content of mycotoxin. However, it takes much time and it is labor consuming. And quite often people would use the method of mixing. So if it is a feed with a high content of mycotoxin, either it should be taken away or it could be mixed with a clean batch so that to reduce the amount of mycotoxins within the permissible limit. And as we mentioned before, biological decontamination is good because it is environmentally safe and it is actually highly specific. It doesn't require any special conditions like high temperature or pressure. This could be seen in physical or chemical decontamination approach. However, biological transformation of mycotoxins should be irreversible, shall be irreversible. It is a very important aspect, and at the very early stages of research, people forget about it. Let us take the example of zearalenone. Here I present two main ways of irreversible destruction that is associated with the loss of estrogenic activity. Either it would be the lactone-reading hydrolysis or ketone carbonyl group cracking. Besides, there are some other microorganisms that transform these OH groups that are in the benzol ring. They could phosphorylate them or do something else with them. However, this is a reversible transformation. You know, in the organism of the body of a person, we have some reversible reactions, and then it's again, we have some toxic things. Some time ago, I searched the information on zearalenone, on deoxyvalenone, and on some other toxins. I found lots of publications on decontamination with different microbial. Per year, there are many 4 or 5 overview and dozens of experimental articles. But when I start looking at them, I see that many articles they do not have any confirmation about this transformation processes, or this topic is covered not in detail. So people just say, yes, everything is damaged and everything works well. However, in fact, if we look at zearalenone, after reading a big amount of articles, I found out that there are just several groups of microorganisms that are able to do such transformation irreversible. By the way, if you are interested, I published the overview on this, on the irreversible degradation. And you know, it is very important at the early stages to understand what you should do and how to do it right, and then you will receive good results. So our study started with the following thing. We were going to check one specific concept, that microorganisms that destroy, everything started with AFB1. So we tried to find them in toxin-producing fungi. The fungi produces toxins, and the fungi produce it so that to survive. And it is surrounded by some other competitors, and toxin is one of the way to to compete all other species. So fungus that live jointly with toxin-producing fungus shall have some specific mechanisms that allow them to make this situation neutral. So we have analyzed 45 fungal strains from the peanut that was contaminated with AFB1, and it happened that 33 strains showed AFB1 degrading activity. Here you can see this is a diagram of activity of these strains. So the smaller the column, the better it is, because it is about residual content of AFB1. And so we were interested in one specific strain because it still had the activity not at the stages of analysis but also after affiliation. So it worked stably at the different stages of cleaning of this active factor. Here you can see the efficiency of its work. On the left side, here you see the numbers. On the right side, you see the percentage. So we see that in 3 days of incubation, half of AFB was destroyed or degraded. And then in future, we conducted some specific procedures on treating, on isolating of this active factor, and we identified that it's exometabolites. It has a protein nature and has some homology to oxidase from Amylaria tabescens. And Miss Larissa Sherbakova was speaking about it. The other very interesting fungi, as far as I remember, it was originated. Miss Larissa, am I right? Was it Gliocularium? Okay. One more fungal straits, Gliocladium roseum or Clonostaches rosea, according to modern classification. In 3 days, it showed almost half degradation of aflatoxin added to the media, and in 7 days, it was a full degradation, almost only maybe 15 or 20% left. This one also shows the exam metabolitis. It also depends on the temperature. The higher temperature was for treatment, the faster this lost its activity. Before, we heard the information that the Gliocladium roseum could do lactanase enzyme that specifically is able to destroy zearalenone through the destroying of lactone ring. So here you can see AFB1 and ZA. So we decided to check this isolated strain, and here you can see that this strain was similar, so it could destroy AFB and ZA. Here you can see the data on lyophilized filtrate, and then in future or later, further, we defined its structure, as Ms. Sherbakova was already telling you about. We created a recombinant producer of the target gene, and actually everything was told about it already. And I want to tell you more details. You know, the use of microbial strains shall be considered as the initial stage. If we find microbial producents, some factors that are able to destroy mycotoxins, it's good because we can scale it up, and it's good that they work in a specific physiological range, no need for some specific conditions, it's environmental safe. However, we always need to take into account that microorganism by itself can negatively impact the processed product. It could produce by itself something toxic, it could just spoil it. And the live microorganisms, they also depend on the ambient environment. You know, enzymes are more stable with regard to the same situation. So, enzymes is the second necessary steps after we identified such elements. Well, basically, this is it. Thank you so much for your attention. Thank you. Thank you so much. Miss Natalia, any questions to Natalia? Please. Could you please use a microphone? I have a question that includes actually 3 sub-questions. Maybe you or your colleague would be able to answer. How to treat it if we speak about industrial scale? Let us take this enzyme and let's take the grain. We have 500 tons. How we will do it? Will we spray it? Will we water grain with it or wheat? Then, what is the concentration, active concentration? Would we use 1 milligram per liter or per ton? I mean, in which concentration this enzyme would be applied? And the third question, again, is it economically viable, and can we scale up this mechanism? Will it be too expensive or will it be affordable? Ms. Larissa, as for the enzyme, I guess you should give the reply. Well, dear colleagues, you might notice that when we speak about some economic indicators, it's not the right question to us. I don't know if it's good or bad. Probably it's worse. Well, now we speak about treatment. These are the grains that are incubated in the solution of clean enzyme. However, we do the treatment with the liquid as well. Why? we use these recombinant enzymes to treat feed, but not for food industry, because here in this feed industry we don't need to have this multi-layer, very expensive treatment system. And all these results that Ms. Natalia was telling about, it's to apply non-cleaned, uncleaned liquid. And in this case, we have the specific percentage of, well, there is a strain. The strain is adapted, and the strain actually has very less protein. So this strain does what you would give it. Plus, it has a rich system of xylanase expression. So the enzymes that would degrade degradable elements. So it increases the nutritional value of some feed. Plus, it is maybe it helps from economic point of view. Speaking about the methods of treatment, nowadays it's not really clear. Probably it is like half a product. Well, we understand that there is no need to soak the grain, because otherwise you have to dry it out. So maybe it should be some kind of half product use. Then as for the amount for the first element, the amount, well, you know, actually the product is efficient and effective, but you know, this is an enzyme. Oxidase activity is a related activity. It's dipeptidase, and for all dipeptidase, it is very characteristic to have low stability. For example, they work with this period, and after some period, it will not be active. You know, in maybe 72 hours, you will have to use a new dose again. So right now we are looking at stabilizing. Maybe we'll use protein engineering so that to increase the stability. But a very high level of expression. As for ZEA, expression is not really high. However, it has the activity, very high activity. In 3 hours, the solution would not have it. I don't know if I was able to answer your questions or not. Let me also tell a couple of words. Can I also add? Do you have a question? I just wanted to say that this is just the beginning, because there are some issues that shall be detailed. You know, we did the treatment, but again, what would be the duration of the treatment? It's still an issue for everyone. So, as Ms. Larissa Sherbakova has just mentioned, that what if not all aflatoxin would be taken? What would be the frequency? How long would enzyme be active? I mean, all of these issues shall still be clarified. But you know, I showed you the table of contamination, and we see lots of cases of contamination. And you know, actually, if we see contaminated batches of grain, it would be economically lost. And again, if this product would not be, let's say, would be quite high about the price, still the economic value of it for big batches of grain would be good. So. As for spraying, there is a specific technology, ultra small volume spray, which allows to use the product evenly for the whole batch of product. As for watering the plants, we have half industrial regulation for the production of protein, and it is quite... Sorry, the person is not using the microphone. Interpretation is not possible. Okay, I think that is enough for the speaker, right? Thank you, Ms. Natalia. And now let us move on to our next speaker and next report. The topic is biologically safe wound dressing based on natural polymers. And Miss Ahmed Karimova Janar, she is the head of the Laboratory of Toxicology and Pharmacology in the National Center for Biotechnology. Hello, dear guests, dear participants of the conference. I understand our day today was very long and it's such a big volume of information, so I will try to be brief so that not to keep you here for quite a long time, because we still have, all of us have some other plans. So let me introduce you our main scientific research within which we did all the activities. So I will speak about biologically safe wound dressings based on natural polymers. And here I want to note that this project is more relevant to biomedicine sector, but maybe due to some technical issues, this report came to this session, to agriculture, and then we decided, okay, we will tell you what we are doing, it might be interesting for you. So, you all know that in National Center for Biotechnology we have 14 laboratories. We are promising, friendly, people work here with decent research activities. And the main goal of our research was to synthesize new modified biological polymer or natural polymer, which is safe, which is non-toxic, and it should be biodegradable. In practices, we use alginate, collagen. There are also some synthetic polymers. After we did a very deep overview, we understood that alginate is economically affordable because the price is quite good. Besides, it has a very good physical and chemical properties. It has very good absorption properties, antibacterial particles. Natrium alginate is known to everyone because we have some face masks so that to have a beautiful face. So alginate is a basis for us. So here we speak about chemical synthesis. By using organic synthesis, we synthesize alginate polymers. We used ultraviolet. I'm not going to speak in details here. It's just the scheme. We received the polymer, and then further, as we have two-stage therapy, we put the particles of silver on the first wound dressing, and on the second it was silver as well as the other element. The first covering has antibacterial effect because it kills, I mean, in theory it should kill the infection. After infection is blocked and no, let's say, distribution of this bacteria, the second layer would be for fast regeneration, so that to restore all of this, so that this wound would be good, let's say, would start to recover. Then we did optimization. There were different other ways and methods, and we received our polymer. We have researched physical and chemical activity of our polymers, so that to prove that the structure complies with, so everything that we planned was received. So the methodology itself, how the chemical synthesis was conducted, we confirmed structure through spectroscopy. So everything was proved. Physical and chemical testing was conducted in Nazarbayev University. Let's say this. I'm trying to find better words to formulate my idea. This project has been conducted with different other integration, science, chemistry, biology, medicine. So we started to do some specific assays. So we identified that it is non-toxic, it is non-harmful. We did all the necessary tests and assays. We did different entity tests and so on. And everything showed us that this dressing works. It has all necessary functions that we plan to have within it. Our next stage was pre-clinic trials. We wanted to see how this wound dressing is going to work with a real organism. And as a subject, we took rats. But again, I want to say we were trying to use our trials in a human way. We didn't use an excessive amount of animals because still we were complying with the ethical rules. We had 4 groups, 5 animals in each. We had a control group and there were some other different components taken into account. And these animals, we did all of the procedures in a human way, so we checked all these wounds through the histological methods, and we looked not only, we looked at different layers of skin, we looked at the formation of the collagen, and at this stage, nowadays, we do the clinical trials or pilot trials based on the National Scientific Center of Traumatology and Orthopedy, named after Batpenov. So they are using our alginate hydrogel dressing on people. Our group is small. We had volunteers. They were informed about all these research features, issues, and they provided, sorry, it's the end of the day, I forgot all the words. So the patient gave us the informed consent, and they use our dressings, and we compare the result with traditional treatment. Unfortunately, we have not the data yet because the work is still under the progress. So again, these are the photos which were before using or during the using of our dressing, but we don't have the photo after. With this, I'm done. Thank you so much for your attention. Any questions? Do we have questions to our speakers, to Ms. Jannar? Please, you can use the microphone. Could you please tell us, the left photo is a traditional, right? Traditional wound. And the right one, this is your alginate polymer, right? No, yeah, this one. This is just the schematic. So this is a wound, then it's the first covering with the silver or antibiotic, and then after the infection is removed, we put the second one. It's argentum or silver and FGF2. FGF2 is your product, right? Yes, this is this your... No, it's one of the components. It's one of the components. What about the burns? Which models? Could it be used for burns? Sorry, it was a question about experiment, how we did the wounds. Actually, we did the cut in the skin in our experiment, and we inserted there staphylococcus or some other infections. This is the way we did our experiment. Could you please tell us what is your product? Is it dressing which is impregnated with gel, or is it the gel that you would just put on your wound and how do you put the substances there or the other components? Thank you for your question. No, our dressing is the gel type wound, and we put our components with the absorption method for 24 hours under the room temperature. There is a specific solution. I mean, all our dressings would be put in a solution, and we wait for 24 hours, and then we do some other tests. Thank you so much. Thank you. Well, thank you. Thank you. So no questions. Thank you so much, dear colleagues. According to our program, we had 9 reports and we have listened to all of our speakers. Now there is the other report from other section, but it has a topic that is related to our section. Maybe it was by mistake put in the other session. The topic of the report: development of new form of biologically active substances for the use and application in veterinary and in medicine. And the reporter is Professor of Russian Technological University, Mr. Yerembetov Kines. Hello, dear colleagues. Let me congratulate you on the 30th anniversary of National Center for Biotechnology. This is a great time, and I think that you will have more development that will be implemented. The person is not using the microphone. Interpretation is not possible. Everything is for people. You know, I presented my report at the medicine session, and the main idea is that we shall not split the science, that this is for medicine and this is for the other industry, for agriculture. I mean, agriculture, medicine, biological science, all of these elements are interrelated and they are tied, and one thing flows into another. You know, when I consulted pharmaceutical companies, I told them, if you are developing new molecule, in your mind you shall always think that it could be used in medicine for medications, it could go to veterinary industry for some specific animal product, or it could go to animal just as an additive, food additive. And in general, we can say that it would go to the food product. So that's why all these developments shall be performed with this idea. The person is not using the microphone. Microphone is switched off. Interpretation is not possible. With the use of supramolecular chemistry, we developed supramolecular complexes for increasing bioavailability of existing or newly developed biologically active substances. I cannot switch it to the other slide. How we can increase bioavailability for the organism? We can use supramolecular complexes with active molecules, or we could use liposomal forms or active molecules on solid lipid media and some others. So supramolecular complexes, these This is like host and guest. We have such complexes in our body, like host and guest. I will try to speak fast and to skip probably some of my slides. For the first time, we received nanostructural supramolecular complexes of the following compounds: 20-hydroxyadisone. We received it from different plants in combination with arabinoglactan. This is from a specific plant, and the ratio is 1 to 10. Then the same compound with Arabic, and we have also derivative rhodanine. It's a synthetically received molecule, selenopiram, and cholecalciferol. It's D3 vitamin with hydroxypropyl. The ratio 1 to 8. Biological availability: 20-hydroxydextrone. We increased 2 times. Derivative rhodanine 6.6 times. This is the increase. Selenopiram increase 7 times, and D3 with hydroxypropyl cyclodextrin. The increase is 5 times. So we did it with the use of our methodology. Physical and chemical properties of selenox. It is not dissolved in water. This is a formula. We registered it. The pharmaceutical substance. This is what we received. Here on the slide you can see the description of the color, and the temperature is indicated. For the first time, the methodology of quantitative defining biological liquids was used with the use of specific spectrometry. And here also you can see the result. Biological availability, we did it using dogs. Our specific approach was used. We used the same dogs so that to exclude influence of cytochromes, because any external part would start to metabolize, starting from intestine and then go into liver. So we did all the mechanism as it should be, and we used the same dogs in our experiments, and we received such good pharmaceutical curves. Actually, we had 2 forms. We did the oily type of form, and we also did the water soluble form. So we did this on dogs, and we have received quite good data on bioavailability. The main principle in the medicine is not to harm anyone, so that's why we are trying to reduce doses. So we use small doses. So we tried to do it in the right way, and we received the indicator 187%, and the complex that used water solution or water soluble substances increased 5 times. Then we used this compound in rats. We did it for rats, and so we see here the main body organs. This is a coefficient of distribution in different body parts. Also, you can see different concentration. In the brain, we've identified it. The barrier was passed. We used in rest, and we found out testicular insufficiency. It was an experiment with the rats. We used Vister population, so we studied different molecules, and let me now skip it and move it and show you the result. So we had intact animals, different groups. And we used the Switzerland medication, paclitaxel, and the results were the following. The number of active form of spermatozoids, the general number, Sertoli cells count, and so this is testosterone. So this is intact animal. This is the spermaris of a rat of reproductive age. Next compound, 20-hydroxytone. It's of vegetation origin. 1 kilo costs 1 million rubles. It's substance, so that's why we decided to do this new complex, so that to reduce the applicable doses and to reduce the cost of price. This is our control group, and here what we see after we introduce this product, medical product. Then we did paclitaxel, as I told you. It's a toxic chemical product. I think I'm going back. Just a moment. 20. The other compound. This is the mean and the particle size. We received 35.3 nanometers. This is the size of the particle. This is UV spectrum of different supramolecular complexes. Biological availability, accessibility is studied in the experiments with rats, and here you can see this red line, and it means that we increased bioavailability and accessibility. Using N15 isotopes, we studied the rate of protein turnover, and we proved that using 20-hydroxycodone, the nitrogen would, the flow of nitrogen would be increased, and the rate of nitrogen of the protein turnover would increase. So this product has anabolic effect. Next, influence of this nano size form to the mass of muscles and the quality of protein. So, and we found out that we can treat the muscle weakness with this, and we also can use this medication for the diabetes treatment. Here we see the mechanism of actions. We see different signal ways. They coordinate metabolism of muscles. On the left side you see the molecules that activate protein synthesis, you see here. And on the right side, here we see inhibition or decreasing the cleavage or decoy. And so you see all signal way go through the specific serine, tronizin protein. Here we see insulin. You see here the rhodamine derivative, lithium, amino acids. All of them activate and increase the synthesis. And on the right side, here you can see the other molecules. So here autophagy. And actually the Nobel Prizes were received for each of this way by people who did it, and all this mechanism actually is done through this system. And here, this is a model of diabetes, and you see that the sugar content decreases. It was before the medication was introduced, the control group, 14, 13. So you see, within 2 weeks we decreased the level of sugar. You know, we used the model of diabetes and we normalized all the indicators in rats. We decreases as this is nano parts, and we studied its impact. Would it influence the chromosome? We see here the share of cells, and we see that there are the same level in control group and in us, and this is a positive control. Cyclophosphamide. And D3 vitamin, we know about it, and using this technology we were able to decrease therapeutical dosage of D3 vitamin 5 times. We know that D3 vitamins, they are all like brought from abroad. Sorry, your time is over. Could you please finish? We see that you are just beginning your report. So this is our experiment with the dogs. We use this methodology of quantitative analysis, and we define the liquid, and this is a complex on D3 vitamin. Here you can see different graphs, so for each molecule we have to choose, and it depends on this. I have just some problems with changing my slides. Here you see. We used sulfasalazine, and I told that using solid lipid substances, we also change some indicators. However, it is toxic and it has some adverse effects, but we were able to decrease this adverse effect. And here we work with upper respiratory organs because the biggest concentration of the product was there. And by using the specific action, we fix the situation. Thank you so much for your attention. Any questions? Are there any questions to our speaker? I guess you're all tired already. Okay, please take your seat. Okay, dear colleagues, we have listened today 10 reports. We even did more than we expected, 9-hour reports and one from the other station. And I want to tell you that all these reports were very interesting. They were dedicated to relevant tasks and issues. Our guests and our guests performed very well, but we have lots of guests today from the Russian Institute of Phytopathology. 30% of our reports were by presenters from Russian Institute of Phytopathology. I don't know about Russia, however, in Kazakhstan we don't do enough activity in this phytopathology. I mean, you don't find, you can count or you can find specialists, but not a lot of them. We have one person from our facility, and she wants to meet you, she wants to exchange contacts with you, and here we will organize such meeting. And I think that today's meeting will be a good foundation for our joint work in future, because we have lots of common problems, and as a veterinary specialist I would like to highlight the report from Belarus State University. I asked him lots of questions during the break, and it's a very promising area, and I'm pretty proud that veterinary science nowadays is at the forefront of biotechnology. And I also really like the report on the development of new forms of biological substances could be used not only in veterinary but in the medicine. So I think that we did a great job. We found new friends, we found new colleagues, and I hope that we will stay in touch in the future as well. So see you tomorrow. Tomorrow we are going to continue. Can I make one announcement, dear participants of the conference? Those of you who are going to the hotel, your bus is waiting for you here at the exit, and if you have the invitations for the gala dinner, you also can use the same bus. Thank you so much.
Достаточно, да.
Institute has been a beacon of knowledge and hub.
До завтра тогда.
поставили здесь один, да? Давайте расскажем. Здесь. Да, я дальше разговаривать не буду. Всем хорошего.